Okay, hello everybody. On behalf of the PHRN, I'd like to welcome you to the next in our Linkage Luminaries webinar series. My name's Felicity Flack and I'm the Manager of Policy and Client Services at the PHRN. I'd like to start by acknowledging the traditional custodians of all the lands we're meeting on today and paying my respects to elders past and present. First, a few housekeeping things. If you've got any questions for today's speakers, please type them in the Q&A box, and you can do this at any time during the presentation. We'll answer as many questions as possible at the end of the presentation. This webinar is being recorded and we will email you as soon as possible when it's available on the PHRN YouTube channel. So to our webinar for today, I'm delighted to introduce Natasha Nassar, who's the Financial Markets Foundation for Children Chair in Translational Childhood Medicine, the Children's Hospital at Westmead Clinical School, along with several members of her team who are going to talk about their work in perinatal and paediatric epidemiology. I'm particularly happy to welcome Natasha today, as we both started our research careers at the same research institute some years ago now. So I'm really looking forward to hear what she's been up to and something about her latest work. So thank you, Natasha, and welcome the rest of the team. Let's get started.
Okay, thanks for having me, Felicity, and thank you for the invitation and the opportunity to present to you today. Yes, we do go way back with Felicity. So I did my postdoc at the Telethon Children's Institute, or the Kids Institute now, back in the 2000s, 2006 to 2008. So yeah, it's a real pleasure. And so I'm going to give you a bit of a story about where I've come from since then. So can you see my screen, Felicity? She's on mute. Okay, I'm hoping everyone can see my screen, and if you can't, then you'll let me know. So
yes, you're good.
All good, great. Thank you. So thank you for today. Yes, so I'm going to be talking about using data linkage for population and translational child health research. And just like to start off by acknowledging and paying my respects to the traditional owners of the land where we're each based today. So today I'm based on the Gadigal, on the lands of the Gadigal people of the Eora Nation. I pay my respects to those who've cared and continue to care for country and any First Nations people who are present here today. So let's see if I can get this moving. Okay, so here's a quick overview about the presentation today. So I'm actually going to give you a bit of a history of child health data linkage and how I've been able to apply this in my research over the last, I guess, 15 years. Felicity's outed me. And then I'm going to just highlight, after the history, how we've basically applied data linkage in various research themes and used data linkage in the research that I conduct at the moment, and basically give you an example, an application of data linkage and how we've applied it to both child health research and its translational impact. And then actually what we're going to do is I'm going to have a few members of my team actually provide and speak to you about various examples of bespoke data linkage that they've been using for various projects in child health research. And then I'll come back on and provide a bit of a summary and a wrap up. So without further ado, so basically our team, the Child Population Translational Health Research team, basically the aim of the work that we do is to use population data to produce clinical and policy relevant evidence to improve perinatal and child health and well-being. So big motherhood statement. And essentially we use an epidemiological approach to basically address 4 key themes as part of our work: to look at the early life determinants of child health and development, to help understand common childhood conditions and injuries, to evaluate value in paediatric healthcare, and to also we also focus on conducting clinical studies in perinatal and paediatric care. Now today I'm going to focus on the first 3 themes of our work, but the 4th theme is actually a really great opportunity where we can apply the findings from our epidemiological and data linkage research back into clinical studies. And we can use the clinical studies to generate hypotheses that then we can then explore in the data. So there's a bit of a virtuous data cycle here that we use in our research, but I'll focus on the first 3 themes. And essentially our vision for all of this research is really about establishing good clinical links and collaboration that we can identify clinically relevant research questions. We're not doing research just for the sake of it because we've got data and we can. It's really about doing, facilitating population health research that has a translational impact. Okay, so before I get started, I have to introduce you to the team because basically this work and this program of work can't be done without my fantastic team, and various members will speak to you today, but really this is really where all the work gets done. This is the real core engine room of the work, and I really want to acknowledge them today. So the research that we do really takes, I guess I want to go back to the core principles, and it really tries to take a lifespan approach to population child research. And essentially it's starting from conception, so pregnancy and birth. And what we're able to do is, what we're really interested in doing is following up babies and infants from the very time they're born into early childhood and adolescence, and now into adulthood as they transition to adult care. And really what we're trying to do is start from maternal conditions and look at how they potentially have a lifelong impact on child health and development. Okay, so that's the lifespan approach, so that's where we're going. So what I thought I'd do now is just start off with the history of how we use data linkage to really take this lifespan approach to our research. So I'm going to take you a bit of a history tour. So way back in 2003, so this is where our group, and when I was based, before I even went to WA at the Colling Institute, and we were a perinatal health research group, and we started with perinatal data linkage. So, and if you look at this figure, you can see the perinatal record. So that's the birth of a baby, and that's a data collection that collects information on every birth in New South Wales, but actually every state has this same data collection. And there's information about the mother's pregnancy, her socio-demographics, her delivery, and some information about the infant. And what we did is we took this perinatal record, and the perinatal data collection, and we linked it to both mother's hospital and baby's hospital data. And what that allowed us to do was not just look at what happened around the time at birth from the perinatal record, but what actually happened to mum and baby at that time. So when they were in hospital having their delivery, how long they were in hospital, what kind of events occurred. The hospital admission data gave us a little bit more information and context around that birth. And then in 2005, actually this is 2006 really, in 2006 the New South Wales Centre for Health Record Linkage, the Cheryl, was actually established. And so this really gave us the opportunity to broaden our horizons in terms of using data linkage to explore perinatal and infant child health. And so the linkage that I've got highlighted on this figure that's circled is just the linkage that you saw previously. So we've still got the mother-baby record from the perinatal data collection, and on either side of those are the mother and baby record that we ascertain from the hospital admission data, so the APDC's admission patient data collection. And so that gives us information about the birth admission and what happens at that time. But then what we decided to do is to use data linkage and then link to additional information about both mums. So you can see to the left of the mother-baby record, and then to the right of that mother-baby record to babies' hospitalizations. And so essentially what that meant was that not only do we have information about the birth at the birth admission, but both for babies in particular that we were interested in their postnatal admissions. And so now what we could do is follow up any babies that might come back to hospital and readmitted after they go home. This linkage also highlights this figure also highlights some additional linkages that we link to ABS death registrations and birth registrations to look at information about subsequent survival of infants, and also to the birth defects register to look at infants born with congenital anomalies. So this next figure basically highlights our next foray and next, I guess, application for linkage. And you can see here that, you know, technology is improving, our access to data and linkage, and our sophistication with data linkage is improving, not just with the color of the slides and the figure, but I guess the detail in this figure really highlights all the different data collections that we now were utilizing in our research. And so this was called the Even More Comprehensive Perinatal and Child Health Record Linkage Project. And this was about circa 2013. So while I'm still based at the Colling, and here we have again the mother-baby record that I've circled, and then now you can see that from the baby record, because I guess that's the focus of the research that I'm really interested in and will focus on today, is if we look at to the right of the mother-baby record, those yellow fields, and not only are we linking to hospital records to identify information and follow up that baby, not just in infancy, but now into child records. So linking, we had linkage up to the age of 6 years. So basically following up infants into childhood and looking at their hospital admissions throughout those first 6 years of life. And then in the blue boxes, you can see all the various data collection we also included as part of this linkage. So we had additional information on these babies that really helped enrich our research in terms of identifying conditions in early life, and then also allowing us to follow up these babies into early childhood and looking at early child development and education outcomes and death registration through and survival. And then this is a figure that I thought I'd show you for, here we are in 2020. So this is basically our new form of linkage, and this is really trying to follow up children, so not just up to 6 years, but really take a broader lifespan approach to data linkage for child population health research. And we call this the Big Mac linkage. And essentially what it does is the figure now changes because we've got so many different data collections. What we've tried to do is show you that lifespan approach in using different health data collections to essentially follow up children across that early childhood and adolescent lifespan, starting from the perinatal data collection at birth, and then following up children through these linkages. So the blue hexagons are highlighting the linkage to various state health data collections, which I'm sure you're all familiar with. The green hexagons are highlighting additional linkage to data collections from various state agencies. And this is where I really have to acknowledge the Telethon Kids Institute, because they were really the pioneers for cross-jurisdictional government data linkages. And, you know, we didn't have any of this when I first returned back to New South Wales after being in Perth. And this really informed this linkage in subsequent years. And then finally, the red hexagons are new linkages that we're now establishing with the Commonwealth data. So to really bring in that information about medication use and primary and specialist care in childhood. So essentially what this has allowed us to do through this data linkage is establish a really rich and, you know, powerful cohort of all births in New South Wales, plus children that are hospitalised aged less than 18 years. And from that study period, so from our different data collections, of course, you know, that they're all collected at different time points, but births are starting from 1994. So we have births from way back in 1994 that we can essentially follow up through these linkages as kids coming in and out of the health system, access different services, go to school, and then all the way up into early adulthood. So essentially, there's about 92 to 95,000 kids born every year, plus a few additional kids that are hospitalized. So it's a cohort, a really powerful cohort, about almost 3 million children. And now that we have, so some of these kids, we have follow-up for them up to 35 years of age. So not only do we have follow-up into early adulthood, but now into sort of real adulthood here. And essentially what this allows us to do, it's a really powerful and really, you know, I don't know, rich database that allows us to explore a whole plethora of research questions, looking at prevalence trends, the burden of disease, to look at the effectiveness and the safety of interventions in clinical care, to look at health service utilisation and costs, to look at early childhood development, school achievement, and overall infant and childhood survival. So look, it's an amazing resource, and we don't take this for granted at all. And look, it really has facilitated a broad range of research questions. And that's what I'm going to show you now, is basically highlight the research themes and the application, how we've used this data linkage to explore various issues related to perinatal and child health research. So the first theme of our work is really looking at the early life determinants of child health and development, and using the data, the various data collections and linkage to various collections to really explore these factors. And you know, I guess what it does do, looking at all these different government agency collections, is really allow us to have a population approach to our research. And what that's allowed us to do is to look at the role of early term births and child development, and that spurred a whole program of research in that work that actually the colleagues are continuing on, and led to you know, a range of different translational impacts in policy, in new indicators, and now new resources for women, and national national plans for managing births around the time of birth, around term births. We've looked at the association between caesarean section and childhood infection, and that's been a fantastic project because that's actually been a multinational collaborative project that's involved people, researchers from Victoria, from Scandinavia, from the UK. So that's a fantastic project that we've been able to contribute to. We've been able to use our data to look at outcomes of infants born with congenital anomalies. We've been able to look at the role of pediatric general anesthesia and cognitive outcomes, and educational outcomes of twin births. We've also been able to look at understanding common childhood conditions and injuries, and we've had a body of work looking at childhood onset type 1 diabetes and diabetic ketoacidosis, and that work has actually led to a whole new program and collaboration with pediatric endocrinologists, and using a clinical database to follow up their kids into early and middle age now. So that's been a fantastic project that's led to really great opportunities after that. We've looked at various injuries in children, sports injuries, falls in infants less than 1 year of age, and the Falls project is actually part of a broader project with a PhD student who's developing an app that's looking at education for parents in that first year, and looking at interventions to prevent falls as children or as infants develop various milestones in that first year of life. We've looked at pharmacological poisonings, and that work has actually been really exciting. It's led to, we've actually been invited to present to the CDC and their Protect initiative in developing policies for labelling and storage of medicines for young children. And we've got a whole new program of research looking at hospitalisation of children with chronic conditions with the K2A, the Kids to Adults Alliance, which is led by Glenn Marshall, Ragulingham and Claire Wakeford, and it's a whole national network of people. So really exciting work. That initial projects that we've just done have led to sort of broader collaborations and research. And finally, we've been looking at evaluating value in paediatric healthcare, and this is really looking at over or underdiagnosis of conditions and treatment and healthcare utilizations and the costing of paediatric healthcare. So we've been collaborating with some great researchers in the Medicines Policy Research Unit at the UNSW with Sally Pearson and Helga Zuega, looking at the impacts and the use of reflux medicines, opioid use in kids, IVI and use in women. And we've also been evaluating paediatric surgery, looking at various types of surgery in children, very common surgeries in children, including tonsillectomies, appendectomies, and orchidopexies, and looking at exploring the timing of these surgeries, the effectiveness, and you know, and where they're being done in terms of location, and looking at centralization versus regionalization of care. And look, with all of these projects, I do have to emphasize that they're being driven by questions that are coming from clinicians, from our collaborators. And, you know, these are not things that we're just making up on the fly. You can see by the diversity that it's really, these are questions that are coming from the coalface in the ground. And we've really worked with various policymakers, clinicians to really drive our research into these areas. So what I thought I'd do now is to actually go in a little bit deeper into one of these particular projects, and sort of, I guess, give you an overview of how the project was initiated, who we've worked with, and how it basically evolves and where it leads to. And this is a project looking at evaluating the newborn screening program for neonatal thyroid hormones, and evaluating the long-term child outcomes of kids with these hormone levels. So I guess I want to start off first by acknowledging Samantha Lane, so you'll hear from her a little bit later, and she actually led this project, so I'm basically just presenting her work here. But essentially, this project really, look, it started off, we were looking at maternal thyroid hormone levels, to be really honest, and then we switched over because there was such an association between maternal and neonatal thyroid hormones. We started to get interested in neonatal thyroid hormones, what their impact was, what kind of influence they had on childhood health, and how they were measured and screened for. So neonatal thyroid hormones are really essential for brain development, for normal growth and metabolism of infants and for children and adults actually. And congenital hypothyroidism is basically abnormal thyroid hormone levels. And it's basically the partial or complete loss of thyroid function that really happens from the time of birth. Now, congenital hypothyroidism is quite rare, but it occurs in 1 in every 2,000 infants. So look, it's 0.05%, but if it's left untreated, it does lead to abnormal growth, delayed cognitive development, and permanent intellectual disability. So it does have really lifelong impacts if it's not picked up and treated. But if it is picked up and diagnosed, the treatment is actually really simple. It's just thyroid hormone supplementation, not just, but it's thyroid hormone supplementations or thyroxine. So essentially, if we can diagnose and treat this early, basically it's being done here in Australia and around the world through newborn screening. It's essentially been able to virtually eliminate congenital hypothyroidism associated intellectual disability in developed countries. So New South Wales newborn screening program, and of course this is replicated as newborn screening program in every state in Australia. It's just run by each of the states and around the world. And in New South Wales, in particular, there's a focus on today, it essentially started in the 1970s. And here in New South Wales, pretty much all infants are screened in the first 48 to 72 hours of life. And it involves a blood sample, so a heel prick of these infants. And that blood is collected and stored on a collection card that you can see here on the right. And the newborn screening test for congenital hypothyroidism in New South Wales is essentially just evaluating the levels. So it's using the blood spot to look at levels of thyroid stimulating hormones, so TSH. And what they do is they look at these hormone levels, and any baby with a TSH level of 20 or above is referred on for further testing. So repeat testing, and if it's a diagnosis, then treatment, so instant diagnosis and treatment. And for those babies that have a level less than 20, there's no further testing and they go on to live their lives. But for us, the question was, well, look, there's these cut points, but you know, how definitive are they? And what happens to children with a TSH level of 9, just below 20, of 19, of 18, of 17? What happens to those children in the long term? Because there's no follow-up for anyone less than 20, we have no idea what happens to these kids. So our study question here was to examine the association between newborn TSH levels and look at the long-term cognitive, education, developmental outcomes of children. And particularly what we wanted to look at was TSH levels just below the newborn screening cut points where there was currently no further follow-up. And we realized that we could actually use data linkage to conduct this study. So if we knew that their newborn screening captures nearly every baby in New South Wales as screened, they have a TSH level that's recorded and stored in the newborn screening database. And if we could link this database to different health data collections and to early child development and NAPLAN information, we could follow up these children through data linkage to look at their developmental and education outcomes by school age. And that's what we did. So here, what I'm going to do is I'm just going to go over the results quite quickly. So here we have the TSH levels on the bottom x-axis of this slide. So essentially what it's showing is the centiles from 0 to 100, the TSH levels over time. And on the vertical axis on the right is the probability of a child scoring below the national minimum standard when they go to school for their numeracy NAPLAN test score. So roughly about 5.5–6% of all kids score below the national minimum standard in numeracy every year. And these are year 3 students, I should just remind you. And these kids usually need further intervention and support to help them get back up to normal levels for their peers. So this is the proportion of kids scoring below the national minimum standard by TSH levels. And you can see from 0, it's pretty steady to the 75th centile. And around the 75th centile is equivalent to about 5 microunits per whole blood. And basically what that means, if you remember that the cutoff is about 20 for newborn screening, most kids, 75% of kids will have a pretty low level of thyroid hormones. But you can see as the thyroid hormone levels increase, the proportion of children scoring below the national minimum standard does increase. Right up into the right-hand corner, these are the levels right up near the cut points. So the very top little star highlights that's that 20th centile. And if we look more closely, here we are. This is the 20th, not centile, sorry, it's the 20 TSH of 20 level of the cut point for newborn screening. And this is where all kids are followed up with a test and for repeat testing, diagnosis and treatment. So it's a really tiny group. So what we thought we'd do is I'll show you this a little bit more closely. So if we look on the left-hand bar of this graph, it's the same TSH levels on the bottom x-axis, but this time we've amalgamated the 0 to 75th centile on the left, and we're going to take a closer look as kids increase in terms of their TSH levels, what happens to that proportion of children who score below the national minimum standard. And you can see here quite clearly that as TSH levels increase from 5, that 0 to 75th centile, the proportion of children scoring below the national minimum standard does increase, and increases to about or just about over 11%. So 11% of kids are scoring below the national minimum standard as that TSH level increases. But what about this little group at the end? Well, let me tell you that this little group at the end are the kids who have a TSH level of 20 or above. These are the kids that are identified from newborn screening, that are referred on for further testing, for diagnosis and for treatment. So you can see that these kids actually do just as well as their peers who are right down here at the 0 to 75th centile. So they actually perform just as well as kids with a low level. But here is the kids just below the newborn screening levels. And you can see there's a dose response increase as the levels just below the newborn screening cut points increase, that the proportion of children scoring below the national minimum standard increases. And this is just the same plot, but what we've done here is we've, so instead of showing you the proportions, these are the adjusted odds ratios. So this is where we've done the analysis, we've looked at the association, but we've adjusted for various different perinatal and sociodemographic risk factors that might influence a child's risk of school performance. And you can see that pattern is still there. As the TSH levels increase, there's that increase in the risk of or the odds of a child performing below the national minimum standard in numeracy, and right at the very end, that last bar showing those kids, no association for those kids who are screened and treated. So these findings actually were, had quite a bit of an impact. They were published in the leading diabetes journal in Lancet Diabetes and Endocrinology. It led on to an international interest in this topic. It's actually quite a big topical issue in newborn screening, and actually Sam was invited to lead an international debate article looking at the pros and cons of what we do with this finding. And what happened with these findings was basically our recommendation was that the newborn screening cut points needed to be reduced. They needed to be reduced to basically pick up those kids just below the screening cut points. And that spurred an international debate where we debated the pros and cons of lowering the TSH cut points. And then following on, because this work was in collaboration with pediatric endocrinologists, with people from the newborn screening program in New South Wales, we actually led to the piloting of lower TSH cut points subsequent to our study, and now actually the lowering of TSH levels. So the highlights from this project was the strengths and the power of data linkage, that we could conduct a population-based study to follow up all children in New South Wales to look at their long-term outcomes, and also the collaboration with key stakeholders. And the other thing I just wanted to highlight with this particular project was that not only the strengths of the data linkage, but there was the strengths and the power of what we call bespoke data linkage. So this wasn't just data linkage of regular government agency data, but newborn screening data that's not part of a regular data linkage, you know, master linkage key. But this is a project, because it was raised by clinicians and a clinical issue, we were able to source this information and conduct what we call a bespoke data linkage of linking newborn screening to different agency data to really explore this question. So that's the real, I think, power of data linkage, that we can have this opportunity to do these types of studies, and then the collaboration with key stakeholders to understand the data, to access the data, but also to have that translational impact. So now I'm going to hand over to a few members of the team to actually provide some more examples of this bespoke data linkage, and how collaboration has really, not only the data linkage, but collaboration has really facilitated us conducting various different research into child health and understanding these issues. Okay, so here we go. I'm going to hand over. I'm going to mute.
Well, hi everyone. Thanks, Natasha. My name is Francisco Schneuer, and I'm a pediatric perinatal epidemiologist, and have worked conducting studies using link data for a little over 10 years, using most of these core data sets and other specialized ones from New South Wales and Western Australia. So today, I'm going to talk about a little piece of work looking at the effects of early childhood exposure to general anesthesia on brain development. So next slide. So in 2016, the FDA in the US released a statement warning about the potential neurotoxicity of repeated or lengthy exposure to general anesthesia in young children. But while there is plenty of evidence from animal studies, there's limited evidence of this association from human studies. And at the time, only one controlled trial and 2 cohort studies had conducted and found a single short-term exposure was associated with increased behavioral problems, but had no effect on child intelligence. But these studies looked at the effect of short single exposure, less than an hour, for one type of procedure, mostly inguinal hernia. So next slide. So what we did is we conducted a data linkage study in children born at term in New South Wales, linking to birth and hospital data to identify procedures under general anesthesia and ascertain the outcomes from the early childhood development assessment and NAPLAN data. So you can see from the figure, we found that any exposure to general anesthesia was associated with a small increase of children being developmentally high risk, so it's vulnerable in 2 or more domains, and scoring below the national minimum standard in numeracy and reading in year 3. This risk was driven by different types of procedures shown in the orange arrows, but these effects were attenuated when we restricted the analysis to children with only 1 hospitalization. So what this tells us is that the evidence is probably, there's little to no effect with 1 short-term exposure, but there is uncertainty about the effect of longer exposure to general anesthesia, with no studies looking at this question due to lack of data. So next slide. So to answer this question, we liaised when we found, after talking with a pediatric anesthetist at the Children's Hospital Westmead, that there was an operating room database called ORSOS that might help us. So this ORSOS collection has information of all procedures conducted under general anesthesia at the Children's Hospital at Westmead, and includes detailed information on procedures and types of anesthesia used, general, local, or sedation. And most importantly, includes the exact start and end times of anesthesia and procedures that could enable the calculation of general anesthesia duration. So next slide. So once we identified this ORSOS database to provide information on GA duration and procedure types, we then use this database to link to the other ones, to the birth and hospital data, to obtain information on sociodemographic characteristics at birth and child conditions, And the outcomes were ascertained by a linkage to the early childhood development assessment at the school entry and UH3 NAPLAN numeracy and literacy test scores. So next slide. So these are a little bit of a preliminary results of the analysis of this new linkage. So overall, we were able to identify about 10,400 procedures under general anesthesia in 3,500 children born in New South Wales at term with developmental assessment information and close to 8,000 children with NAPLAN records. So in the left, we have figure I present the distribution of time under general anesthesia, highlighting that most children had short exposures of less than an hour, but there is a large proportion exposed to more than 1.5 to 2 hours under general anesthesia. So this allows us to explore more lengthy exposures. And on the right, you see a scatter plot of each child's duration plotted against the NAPLAN scores on a continuous scale. And as you can see, there is a mild downward linear association of decreasing NAPLAN numeracy scores with increasing time under cumulative time under general anesthesia. So the next step that we're going to do in this analysis to explore the association further, we'll look at different cut points and use different options to select comparison groups, attempting to isolate the effect of general anesthesia. This will involve use techniques such as propensity score matching, looking at unexposed siblings, and exploring the effect of counterfactuals where it is possible, which we'll be looking at children admitted for similar diagnosis in early childhood but without general anesthesia. So next slide. And so this study has a number of clinical and translational outcomes. First of all, we find this will help identify whether there is a threshold for time under general anesthesia that is safe for children in early childhood. Also inform clinical guidelines about the potential risk of lengthy exposures to general anesthesia, and help clinicians in decision-making and parental counseling about when procedures might be avoided or delayed. And to finish, this has been a collaboration between pediatricians, pediatric surgeons, anesthesiologists, educational psychologists, biostatisticians, and us epidemiologists. So that's all I have for today. And thanks for listening, and handing over to Diana.
Hi, everyone. Okay, the host is unable to share my screen, so don't know if that can be fixed. Okay, well, oh, there we go. Okay, so hi, my name is Diana. My background is actually in nursing, midwifery, and clinical trials, now transitioning to population health. I've been working with Natasha's team for at least 10 years, I guess. And so today I'll be presenting our study, which is assessing the outcomes of children admitted to pediatric intensive care units in New South Wales. Now in 2015, approximately 2,700 children were admitted to a PICU in New South Wales. And the high mortality and morbidity of these children not only place a disproportionate burden on healthcare costs and services, but also results in long-term implications for the individuals. And although advances in science, technology, and systems of care have improved the survival of children in hospital, there is a potential that more children go home with a decreased quality of life and are more likely to have chronic complex conditions, resulting in potentially later deaths. However, the long-term outcomes of PICU survivors has not been evaluated in order to determine how best to care for and improve the quality of life for these kids. So therefore, the aims of this study are to use a data linkage approach to determine the long-term health, developmental, and cognitive outcomes of children who have been admitted to a PICU. And secondly, to determine healthcare resource utilization among children with PICU admissions and estimate the cost to the healthcare system. So recently, we conducted a study evaluating admissions to PICU and subsequent readmissions to hospital. Now, this study has just been accepted for publication, but it used hospital admission data to evaluate outcomes which, although being adequate for our study, it lacked the clinical details of care provided in ICU, making it difficult to ascertain specific treatments that children received. So other limitations that we discovered when analyzing the data included not having temporal information of ICU versus hospital care, and also not being able to determine the severity of illness scores, which is useful for calculating risk-adjusted mortality and determining a change in admission thresholds. So to overcome these limitations in this current study, we're including the Australia and New Zealand Pediatric Intensive Care Registry, which is a data set that will give us the detail required to adequately assess our outcomes. Now, the ANSPIC provides a comprehensive picture of pediatric ICU admissions in Australia and New Zealand, and its main objectives are to collect comprehensive data reflecting PICU activity, enable monitoring and benchmarking of PICU performance, and to promote and facilitate research into critical illness in children. So this data set will not only give us information about diagnoses and reason for ICU admission, but give us that much needed detail about clinical conditions and interventions and procedures performed during the PICU admission, importantly including start and finish times. We'll also be able to determine unplanned readmissions to ICU rather than just hospital readmission, and be able to use documented physiology data to calculate the severity of illness scores, which was lacking in our previous study. And as well, we'll be able to receive additional death-related fields. So now this project uses a lot of data sets in order to collect the outcomes that we need. So it's collecting state health data sets as well as Commonwealth supported ones. And these in themselves will provide a significant amount of data for analysis of our study outcomes and exposures. Next slide. However, the addition of these external data sets will just enable us to fill those gaps that are difficult to fill using just the health data only. Using ANSPIC to link the neonatal, educational, and development data sets with our standard ones will help us to assess additional exposures that may influence our outcomes and just provide a richer and more in-depth level of clinical detail not found in the standard data sets. So in regard to implications and future directions, there is a need to understand the impact of PICU care and interventions on health and well-being, to map pathways of care, to inform the development of a long-term follow-up clinic for PICU survivors, and finally to assess healthcare utilization, costs and resources, particularly in determining the benefits of centralized versus regional care in the future. This study is being conducted with collaborators from the Sydney Children's Hospital Westmead, the Boston Children's Hospital, and the University of Sydney. So now I'll hand you over to Sam.
Okay, you've got me. Thanks, Diana. Hi, everyone. My name's Sam Lane. I am a perinatal epidemiologist as well. I've worked with linked data, I think, since about 2016, which makes it about 15 years, which is a bit frightening. But I'm going to talk to you today about a project looking at congenital heart defects. Next slide, Natasha. So just a bit of a background about congenital heart defects. It's the most common congenital anomaly. It's about in 8 to 10 children per 1,000 births have a congenital heart defect. And so the term congenital heart defects is an umbrella term and can include a lot of different diagnoses, from simple heart defects such as ventricular septal defects or holes in the heart, to complex defects which may need to have surgery, many surgeries across a child's life. So about a third congenital heart defects need a surgical intervention. And over the past 20 to 30 years, there's been really big advancements in treatment. And so now about 85% survive to adulthood. So we've done a bit of work on congenital heart defects, but in the past we've used hospital data to identify them. And we've known from validation studies of hospital data that procedure codes are recorded more reliably than diagnosis codes. So in the past we've used cardiac procedure codes to identify these kids, but our past work was in collaboration with paediatric cardiologist Gary Scholler and paediatric cardiac surgeon David Winlaw. And while we were working with them, they told us that they had a clinical data set that that we could link. So Natasha, yep, their clinical data set was the Kids Heart Research DNA Bank, and they've been collecting data on individuals, about over 3,200 individuals since 2002. So in this data set, there's a lot of extra clinical detail that isn't in the hospital data, such as kind of specific diagnoses, diagnostic test results, family history of cardiac defects, and also other syndromic information. But another thing that they've been collecting is DNA samples. So speaking to the clinicians about linking this data set and projects that we could do, that they had this clinical question that, Natasha, if you could click it, that they were wondering, why do some children have poorer cardiovascular outcomes compared with others, despite having completely the same diagnosis of congenital heart defect and the same surgical procedure? So perhaps was this a genetic variation that can explain this difference between exposures? So this has led to a collaboration with Victor Chang Institute and Children's Hospital at Westmead to look at this research project, and we've been funded by NHMRC Synergy Grant. So the aim of this project is to examine whether poor and good cardiovascular outcomes in congenital heart defects patients are associated with genetic markers. So to do this, so our part of the project is to find the outcomes. So we've linked the Kids Heart Research DNA Bank with Registry of Births and Deaths to find survival, also hospital admissions to find any diagnoses of poor cardiovascular outcomes. And then to find the good outcomes, we just need to make sure that these kids are still residing in New South Wales. So we've linked it to NAPLAN and the emergency department. So as I said, they have about 3,200 kids, but so now once the poor and the good outcomes are identified, we then need to send the DNA samples to the Victor Chang Institute for whole genome sequencing. But whole genome sequencing is very expensive. So they only want 450 children with good outcomes and 450 children with poor outcomes. So this gave us a bit of a challenge. We then have to re-identify the individuals to find those specific DNA samples. So between us and the Cheryl and Ethics Committee, we've worked out these methods to still have this the separation of data principles. So no one has identifying information and also has outcome information. Okay, next, Natasha. So the progress at the moment, we've linked the data, and so I'm kind of looking at the outcomes at the moment where we're doing it by, as I said, that it has diagnosis information. So we're doing it by the congenital heart defect type and also age of surgery. And this is having a lot of consultation back and forth with the clinicians to define exactly what are good and poor outcomes for each of these different diagnoses. And so the plan is hopefully we'll be able to identify these kids by the end of the year, and then we can send the DNA samples to the Victor Chang Institute next year to begin the whole genome sequencing. And they'll then analyse that to see if they can associate any genetic variation between good and poor outcomes. So in terms of translation, we work closely also with, there's a genetic counsellor in our research group. So this can help with genetic screening and counselling. It can also identify patients that are possibly at poor risk of outcomes, which may help in looking at their treatment options. And also it's kind of a step towards personalised medicine, which is different people may have different treatments based on genetic information and also personal characteristics. So this is a great collaboration between kind of paediatric cardiologists and cardiac surgeons, also the lab-based people at Victor Chang, and genetic counsellor. And it just shows, I guess, that you can go like one step further from data linkage to perhaps explore more into the causes of these things. Okay, I'm going to pass on now to Simon to speak about his project.
Thanks, Sam. So hello, everyone. My name's Simon Padget. I'm Natasha's PhD student, and in my day job, I'm also a paediatrician working here at the Children's Hospital Westmead in Sydney. And so this project focuses in on one common chronic childhood condition, cerebral palsy. Next slide, Natasha, please. So CP is a common condition impacting an estimated 40,000 people in Australia. CP is defined by multiple pathways that can cause disruption of processes in early brain development and result in a disorder of movement and posture. CP as well is a very diverse condition, and as well as the physical disability associated with CP, many children also have other health conditions or associated impairments as shown in this slide. And as with other chronic childhood conditions, much of CP management is performed through outpatient clinics designed to detect problems early and provide evidence-based interventions to improve outcomes. I work in one of these clinics. So children from all across the state come to my clinic, and it's important that those clinics are accessible and equitably accessible. So for this project, we're interested in looking at patterns of outpatient attendance for children with CP and how factors such as living in regional or remote areas might impact on access to these important services. And we also want to consider how differences in access to outpatient care can impact on health outcomes, and in particular the unplanned use of other hospital services such as hospital admissions and emergency department attendances. To do this, we're using linkage of a CP register with the health administrative data sets that we've already discussed. But I just wanted to point out that these data sets also involve a newly available data set, which we call the NAP, the non-admission patient data set, which data collection, which also details outpatient clinic visits at public hospitals throughout the state. And as well as that, we're using hospital admission and emergency department data. So as I've said, this project used linkage of routine health administrative datasets with the New South Wales ACT CP register. The CP register is a population-based register containing details that are highly representative proportion of the New South Wales and ACT people living with CP. The register offers substantial value for this project. Firstly, it contains clinical details of CP severity and comorbidities that aren't available in health data sets, meaning that we can explore the impact of these factors on healthcare utilisation and adjust for these factors in our analyses. And secondly, sorry, just on the next slide, studies in other jurisdictions have shown that CP is not well captured in health administrative datasets based on ICD codes. For example, this study in Canada suggested that only 2/3 of children with CP are captured in health datasets, with a bias to those who are born who have more severe CP who are born preterm. So we've got some early results from our analyses from linking these data sets, suggesting that, for example, in adjusted analyses, that not attending scheduled outpatient appointments at children's hospitals is associated with socioeconomic disadvantage, an older age of the child, and previous non-attendance. And we're hoping that these results and others will be very helpful to inform changes to our model of care for CP, helping us develop systems and resources that that monitor and support access and improve the use of e-health technologies and telehealth to support children and integrate better with their local health systems to enable care close to home. So as with our other work, this is very collaborative work, and I'm very grateful to our collaborators from the New South Wales ACT CP Register and also from Sydney Children's Hospitals Network. I'm also grateful for the funding that we've received to support this work. Thank you guys, and I'm handing you back to Natasha.
Okay, here I am. I'm back. Okay. Working. Let's have a look. Yes. Okay, so I'm just going to quickly summarize and wrap up now. So thank you everyone, thank you team for just giving you a bit of some highlights about the various work that we've done, really highlighting, you know, this lifespan approach using data linkage to understand and conduct child population health research, and moving just from beyond just the standard data linkages that you can get from government and administrative data sets, but to basically be able to incorporate these bespoke data sets to really enrich, you know, the analysis and our understanding of childhood conditions and pediatric care, and the value add that it really brings to us understanding and improving child health. And so this is, you know, in terms of future directions, I'm just highlighting here the various different bespoke data collections we're going to include in future work. And essentially to really highlight that the factors for successful research, data linkage research, is to establish good clinical links and collaborations that are multidisciplinary, to go use this data to go beyond descriptive research, to have a real clinical impact and translational effects, and these will have clinical and policy implications, and having clinical and policy champions that really can move the research into the clinical and policy realms. And essentially, these findings can have multiple translational strategies. We're not just publishing papers, we're not just trying to increase our track records, but really making a difference to clinical guidelines, to policy, to health, improving the health system and financing, and also improving consumer education and decision making. So I just want to finish by highlighting and just all the various collaborations that we have across the university, across Children's Hospital at Westmead, of other universities across the state, interstate and internationally. And this work can't happen without these various collaborations. We can't work in isolation as researchers, but really by working with these others and these various networks has really enhanced our research and really ensured that not only do we have a clinical impact, both local, nationally and internationally, but that we also improve the lives of children and young people, you know, from the very start of life, to ensure that they have a healthy start of life and that continues across the lifespan. So thank you for your attention. I'm sorry I've gone a bit over. I just want to finish by acknowledging the Ministry of Health, the Centre for Health Data Linkage, the various custodians that provide the data for this work, the Population Health Services Research Ethics Committee, who are great, provide a great service and a great, you know, great support to our work, the various funding bodies that support all of this work, and we can't, you know, it's not possible without them. And finally, to acknowledge the wonderful team that not only that you've heard from some of them today, but the rest of them who this work can't happen without, and that I, you know, admire and value and yeah, want to acknowledge today. So thank you for your time, and I'm happy to take a few questions. And if not today, you can always email me. My email address is here. You can contact me later on if you need. Thank you.
Thank you, Natasha and team. That was a fabulous presentation, which really demonstrated the breadth of work that you can do with linked data, and definitely demonstrated what the PHRN's vision is to improve the health and well-being of all Australians. So that's a lovely demonstration of how that could be done. We've got a few minutes left, and we've got a couple of questions in the Q&A box. So I think we will try and get through those. So if all of our presenters can turn their cameras on, that'd be great, so everybody can see your faces. The first question is, where is the linkage done? And in particular, who's linking the Commonwealth PBS and MBS data?
Okay, great questions. And well, the PHRN is a great facilitator, but actually a lot of the linkages are done by the different state data linkage units. So in New South Wales, where we're based, it's the New South Wales Centre for Health Record Linkage, or the CHRL. And so it's done independently. It's not done by us at all. We apply for access to all these different data collections, and we get approval from the custodians, we get ethics approval, and then all of these linkages, the data is sourced by the CHRL and then linked by the CHRL, and they provide us identified data to conduct our analysis. For Commonwealth data sets, it's a separate process. We apply through the AHW, and essentially the same thing. There's an application, an ethics process, and they conduct the linkage with the Commonwealth data collections, and then actually they put that data in the shore environment for us to conduct the analysis.
And I should just say that the AHW, the CHRL and the shore are all funded by the PHRN. They're all part of our network. So Very good. Second question relates to the first two studies that were discussed that involve NAPLAN scores. NAPLAN scores are dependent on a whole range of factors. When associating TCH levels or anesthetic exposure to NAPLAN results, were the confounders controlled for?
That's a great question, and it's, you know, that's a big part of our work. Of course, we have to, you know, there's so many factors that drive, you know, childhood development, whether it's cognitive or behavioral, and, you know, and that's, I guess, the value of having all these different data collections that we can ascertain information about perinatal factors, you know, being born premature, being born, you know, small for gestational age, socio-demographic information of the parents, mum's age, whether she smoked in pregnancy, the family dynamics and SES, you know, all these issues are so important and relevant in terms of a child's outcome, and that we need to take into account. So not only are we adjusting for all these various factors, we're also using these various statistical approaches to also try and really tease out the effect from all the confounding and then the other variables that are going on. And we're using techniques like propensity score matching, like looking at counterfactuals, you know, using different mediation analysis to really try and tease out that question. So you're absolutely the person asking the question. Thank you for that, because it's such a complicated question, and it's not just a cause and effect, but we have to take into account the bigger picture, and that's what we're trying to do with the data as well as our analytical approaches.
Okay. And we've just hit the hour, so I think we'll leave the questions at that, and I'll say thank you to all of you once again. That was a really fascinating presentation, and we're delighted that you were happy to do that for us. And I just need to quickly share my screen to remind people that we have our final Linkage Luminaries webinar coming up next month, which is Dr. Matthew Jose from University of Tasmania, who's going to talk about kidney disease. So if that interests any of our audience, please make sure you register, and I'm sure that will be an equally interesting presentation on a very different area of disease for everybody. So thank you once again, and goodbye to everybody, and we'll see you next month, hopefully. Thanks, everyone.